going to get back to the immune system today, and we're going to talk about adaptive immunity. So this is part 2 out of 3 parts for the immune system. We're going to be talking about immune disorders next day. So today, all about antibodies, and vaccines, and a bunch of other things. So let's get into this. I think last day I had made on the whiteboard something that looked like this. And I mentioned that there were kind of 3 parts or 3 lines of defense for the immune system. So last day we talked about innate immunity, which is non-specific but very fast. And we can't have an immune system without that. If we were to pick any part of the immune system to keep, innate immunity would be it. It's actually preventing most of the invaders from getting into our bodies. So that included 2 lines of defense. First was physical barriers, so the skin and the mucosal membranes and all their secretions, such as salt and sebum and lysozyme and things like that. And the second was cellular and chemical responses, so that includes all sorts of different cells and responses, such as inflammation and fever. So today, we're going to talk about the adaptive immune system, which is specific, which means it is targeting a very specific pathogen or a very specific toxin or something like that. And it also has memory and is a very, very strong type of immunity to have. And we want to keep that one too. So this is going to involve cells called lymphocytes, which are sometimes called B cells and T cells, and antibodies. So I wanted to kind of introduce a few things and make a few definitions before we really got into the adaptive immune system. First thing I wanted to talk about, I'm just making a mess here at my desk. I wanted to talk about kind of some of these features of the adaptive immune system, meaning these are things that it can do. So, and they're kind of definitions as well. The first one is specificity. And so what do I mean by specificity? This is where the immune system is going to recognize something specific. So we'll say that, recognize a specific antigen. So what do I mean by antigen? I'm going to talk about that in a minute. An antigen is something recognized by the immune system. It could be a pathogen, it could be a toxin, it could be some sort of surface feature on a virus, those kind of things. Inducibility. So what does that mean? It means that the immune system will get turned on, I guess, when it's needed. So immune system, by that really I just mean the immune cells are activated in response to pathogen. There we go. Hopefully you can read my writing okay. Number 3, clonality. So what do we mean by clonality? A clone is basically a genetic copy of something that's genetically identical. So this means that our active cells are able to make clones of each other. So we can say that active cells are able to duplicate themselves, are able to duplicate themselves. There we go. Okay, number 4, no response to self. Hopefully that one makes sense. We don't want our immune system to attack our own body. So we can say that. The immune system should not react to its own body. Okay, one more, memory. So again, hopefully that one makes sense, meaning that this part of the immune system can remember and react to future infections. So we'll write that. The immune system can remember, and react. I'm not actually going to say more quickly and stronger. Quickly and stronger to future exposures to the passive pathogen. So the adaptive immune system, often when people think about the immune system, this is often what they're talking about. Lots of people have all sorts of experiences, maybe getting an infection or getting a vaccine and then knowing or hoping they're protected in the future. People don't always think about the innate immune system. Like I said, the innate immune system is very, very, very important. And both of these parts of the immune system do interact with one another. So now I want to kind of talk about the components of the adaptive immune system. So also 5 parts, and tell you a little bit about what these things are, and then we'll talk about how the adaptive immune system works. The first one there you can see is the lymphatic system. So not everyone seems to even know the lymphatic system even exists. Hopefully you have talked about a little bit in your anatomy and physiology class. Basically, it's a secondary circulatory system in your body. So we all know about the primary circulatory system carrying blood. This is another system with little vessels, and they're not carrying blood, they're carrying lymph. And lymph is basically plasma and waste from the blood, but also contains all sorts of immune cells. And some of these areas around the body where there's pockets of immune cells are called lymph nodes. And if you've ever had an infection where they swell up, you may have noticed that. Sometimes when you go in for a checkup, if you're sick, a general practitioner, they may feel under your chin, or sometimes in your armpits, places where lymph nodes are, to see if they're swollen. When I was in grade 10, I had mono, and I remember my lymph nodes in my neck were so swollen up I could see them when I looked in the mirror, and they were kind of painful. They were so swollen up so much, my immune system was going crazy. Someone has a question about why the immune system sometimes does not retain immunity to things. That's kind of a mystery. I don't think we actually have a good answer to that. We'll talk about a little bit when we get to the vaccine part here. But there's a lot we don't understand about the immune system. Some things, like if you were vaccinated against smallpox 60 years ago, which none of us, I don't think, are that old here, you probably still have a very strong immunity. Immunity to measles is very, very strong, lasts a long, long time. Immunity to something like cholera seems to last a year to 5 years, and the person making the comment is asking about immunity to rubella, which I'm not as familiar with. But pretty much there's always some people, whether it's a genetic quirk or whatever, that vaccines don't seem to hold to. Some, like the measles vaccine, is highly effective, like 98, 99%. Pertussis is more like 80%, which is why we have pertussis outbreaks, because for some reason about 20% of the population don't get good immunity to it, and we don't really understand it very much. But very good question. So what else do we have here? We also have antigens. So what are antigens? Antigens, of course, are the invaders, right? These are the things we're trying to get rid of. Antigens are usually some sort of macromolecule, so a protein or a lipid or a glycolipid, sometimes carbohydrates, sometimes other molecules such as penicillin. Could be an antigen if you're allergic to penicillin. And usually these are things that are found on the surface of the pathogens that are trying to infect us. So some examples here, you can see we might have a surface polysaccharide on a bacterium, or maybe a glycoprotein on a virus, or maybe a toxin itself. So some toxins or toxoid vaccines we have, for example, we have, if you ever had a tetanus shot, you're actually getting a vaccine against the tetanus toxin, not the bacterium, but the toxin itself. So we'll talk a little bit more about that too. So number 3 are antibodies. So antibodies you're going to probably see in my notes a lot. They're abbreviated as AB. Sometimes they're called immunoglobulins or IG. So immuno, of course, is for the immune system. Globulin is kind of a term meaning a soluble protein. So you'll see both of those terms. I usually say the word antibody, but some of them do have abbreviations. Like you might see an abbreviation that looks something like this, IgG or IgM. It's talking about different types of antibodies. So what is an antibody? It's a protein. It's kind of complex. If you take a look at my little cartoon there, it has 4 strands. So what I'm doing here, I'll draw them on here. We've got 1, 2, 3, 4. 2 of the strands are kind of short, 2 of the strands are kind of long, and they're held together by something called disulfide bonds, which is not that important. But what is important is these antibodies. First of all, you can see they are Y-shaped molecules, and secondly, they have a couple of things I want to point out. One is these antibody binding sites. So they actually have 2, by the way. So I should have circled that one as well. And this antigen binding site is what is going to recognize the pathogen. So the glycolipid or the toxin or whatever. So this is what's recognizing the foreign invader. So this is good. We can our body can generate these things. Sometimes it's called the variable region, meaning that we have many different possibilities there. Because think about all the different possible things you could get infected by or vaccinated against. You know, you could be mono, could be chickenpox, could be maybe SARS-CoV-2. You're making these things like crazy. So there's many possibilities. The other part is the called the constant region. So that's kind of down at the bottom, and that's what's recognized by immune cells. So it's kind of like the adapter. So this is the part that's going to connect and bring the pathogen over to immune cells. So I think I have some slides here on antibodies to show you some of the things that they do. But the main thing to know is that they recognize something called antigens, right? Which are the invaders. Sometimes you might see the term epitope. I don't have that in my notes, and I won't ask you what that is on a test, but that's something a little bit more specific than antigen. It means the exact amino acids or something like that that is connected to. So you can see there's some antibodies attaching to different parts on a bacterial cell, and there's some also attaching to a protein as well. So let's talk about what antibodies do, okay? They bind antigens, but so what? Turns out that that's really important because this is going to lead to the destruction of those pathogens. And I have a slide for each of these, so let's take a look at them each in a little bit more detail. First one is something called opsonization. So opsonization is kind of a fancy word that means something is getting bound and targeted for phagocytosis. So you can see there's my bacterium. It gets labeled by an antibody, and then a macrophage, or that's a phagocyte that will kill something by phagocytosis, recognizes it. So it's going to bind to that constant region kind of over here, going to bring it in by phagocytosis, and it's going to destroy it using digestive enzymes in a lysozyme. So that's kind of one of the main things that antibodies do, is they're going to promote phagocytosis, and this is called opsonization. So another fancy word to learn. Sorry, another word we don't need, but I didn't name it, so I guess we'll have to live with it. So as we also mentioned, some things are going to be too big to be killed by phagocytosis. So we can have the non-phagocytotic killing, also called cellular toxicity, cytotoxicity. So you can see it's got a big fancy name, antibody-dependent cellular cytotoxicity. So this is like that kiss of death thing, right? It's being targeted, and it's not able to kill it by phagocytosis, so it gets targeted, and a lymphocyte, this NK here stands for natural killer, comes along and basically injects it with some toxins, and the toxins do things like burst holes in the pathogen and those kind of things. Somebody's asking if antibodies get broken down like the bacteria when they're phagocytized. You know what? I'm not 100% sure with that, but I think that they do because they're getting shoved into a lysosome with all sorts of digestive proteins. And don't worry, your blood is going to make lots of antibodies and result in infection. So we have phagocytosis, that kind of killing. And we also have the non-phagocytotic killing, which is, like I said, delivering toxins to the pathogen. So there were a few other things. Another is activate the complement system. So I didn't want to get into the complement system a whole bunch last time, other than to tell you it was a way to enhance the immune system. This was the definition I gave you before last day. I said it's a bunch of proteins, it's about 30 proteins, and they can trigger other immune responses such as inflammation and fever, opsonization, so there's that word, they could trigger phagocytosis or other types of killing, right? So the complement system, like I said, it recruits immune cells, and it's very good at that, and then the immune cells do what they do, which is killing the pathogens. Another thing is agglutination. So we talked about agglutination way back in, I think that was about topic 10 or 11 as a diagnostic technique, right? You add these antibodies and you look for clumping. Well, the immune system can do that too, right? It's going to clump things. And what's the point there? Well, it turns out that things that are clumped, bigger, they're more visible to the immune cells, and more likely to get killed by phagocytosis. So the agglutination is really leading to phagocytosis in most cases. We did talk about agglutination, like I said, as a diagnostic technique. I can't remember whether I shared this slide with you or something different. This is a method for determining someone's blood type. So you can see the blood type test, you know, they prick the person or get a little blood sample. You put a little bit on a couple of drops on a glass microscope slide or a dish or some sort, and you have two solutions. One have antibodies to the A antigen, and one have antibodies to the B antigen. So you can see they're called anti-A or anti-B antibodies. So if you add the anti-A antibodies and the person has A blood type, it's going to bind to the A antigen, and you're going to get the agglutination or the clumping. If you add the B antibodies, there's no clumping. So in that case, the person is A type. Similarly, if you have B type blood, the clumping will be observed with the anti-B antibodies and not the anti-A. If you have AB type blood, you'll get clumping with both types of antibodies. And if you have O type of blood, you don't have the antigen on your surface. It's actually a carbohydrate. You don't have that, and so there's no clumping. So it's a pretty easy test. I know there's A, B, and AB, and O negative or positive, and that's actually a different antibody. You can look that one up if you want. I think it's called the D antigen or something like that. So something else that antibodies are really good at in some cases is neutralizing something. So what does that mean? It means they can kind of coat the virus or coat the toxin, and then it's not functional anymore. And we know this is very effective because, as I mentioned, the tetanus toxin is one of the most potent toxins on the planet. And if you have your tetanus shot, you can produce enough antibodies that you have zero effects whatsoever. They've done these tests in animals where they inject toxin in the animals when the antibody titer in the blood is high, and there's no biological effect. So it's very, very effective. And of course, usually also this leads to often opsonization, which is phagocytosis as well. So there's our antibody functions. You can see down at the bottom, usually this is leading to phagocytosis or the other type of killing, the cellular cytotoxicity type of killing. And that's kind of the point, right? We want to clear our body of the pathogen and kill it and make sure it's not causing us any harm. So just to make things a little bit more complicated, there are many types of antibodies, actually 5 that have been classified. I'm not sure why it's not A, B, C, and so on. I have no idea. They're given those fancy names. You can see IgG, IgA, IgM, IgE, and they're all just a little bit different in terms of how they're put together. As you can see, the IgM is really fancy. It has not 2, but 10 binding points, which makes them really, really good at kind of the early part of an infection. So they all have slightly different roles. So if you take a look IgG, it says it can do all those things we talked about. So it can be involved in opsonization and complement activation, all those kind of things. But it's important because it can actually cross the placenta. So if you're pregnant or if you're a fetus, there's that that benefit going to the child, getting some of its mom's immunity and antibodies. Because of course, when the child is born, its immune system is very, very weak and undeveloped, and it takes months and years to develop. So it's good to have a little bit of that floating around your blood to protect you against some diseases. IgA, kind of similarly, actually can go across and get put into the breast milk. And so another way that, this is one of the things that, they're always promoting breastfeeding, which is probably a good thing. But one more reason to promote it is that the mother can provide some immunity to the child. IgM, like I said, is kind of one of the early released antibodies, and it's really sticky because it has all those binding points. And so it's very, very good at kind of just, getting released by the cells and hopefully clearing the pathogen. IgE is involved in allergies, and it's also involved in anti-parasites. So by parasites, I mean worm kind of responses. And so, as I mentioned, those parts of the immune system are kind of intertwined. In that, if you're getting exposed to pathogenic worms, then you're probably not having a lot of allergies because that part of your immune system is doing what it's supposed to. But in modern kind of industrial societies, we don't tend to interact with pathogenic worms so much. And so that part of our immune system is kind of trained and ready to go, and sometimes it reacts to the wrong thing and we get something called an allergy. So we'll talk about allergies a little bit more next day. And then the last type is IgD. And as far as I can tell, we don't actually know. I think there's some hypotheses around it. The immune system is very complex, so there's going to probably be a lot of surprises that we will learn over the next few years. Someone says a healthy human has all 5 types of these antibodies. I believe so. I think IgA is not only just in the breast milk. It's also found in the mucous membranes, is my understanding. But yes, there are some people that have immune disorders where they're missing one type or something like that. And usually the other types do compensate, but they might be more susceptible to certain types of infections. So we talked about antibodies as something very, very useful for all sorts of diagnostic techniques. So the ELISA was a diagnostic technique that uses antibodies. And so I had mentioned that these ELISA tests are usually done in these little well plates that, they have 96 wells, and you can do a whole bunch of samples. You put here's kind of the procedure. I'll just kind of go over really quickly. The sample would be put in a plate. And the sample could be saliva or blood or some other specimen, and it coats and gets stuck to the plastic. And then you're looking for something. So let's say this is an HIV test. That would be a blood sample usually, and so they would have an antibody against not actually the virus in HIV. In fact, the virus is usually found in such small quantities, it's easier to test for the person having an immune response against the virus. So we'd have an antibody that would detect an HIV antibody. It kind of sounds weird, but it works really, really well. It kind of gets washed and cleaned, and then there's a secondary antibody that has an enzyme on it, and that enzyme reacts with the substrate to produce a color. So this is kind of like this cascade we were talking about with the complement system, is that you have one antibody binds 2, and then it binds the enzyme which reacts with a lot of substrate. So it amplifies the signal and makes it very, very, very sensitive. So we had also mentioned before, also there's a sample of an ELISA assay in a multi-well plate. You can see there's some negative and some positive results. And I had also mentioned that there's all sorts of rapid tests that are kind of based on the ELISA principle. And so here's a rapid HIV test, very similar to a pregnancy test, in that there's a little place to put a sample, whether it's blood or urine or something else. The sample is going to diffuse, bind antibodies, and you're going to see a reaction. So if you have two bands, you can see the control band and the test band. That means you're positive. If you have just the control band, that means the test is working and you're negative. Other results are invalid because the control band should always go, otherwise the test is not working properly. So I think if you want to review that, you can go back to those notes from before. There was a little video I shared with you to kind of show how these work. It's kind of really cool design. Somebody's asking, should we know what each antibody does for the final? I am not, no, I will not ask you what IgM does versus IgG or anything like that. There's only so much I can ask you, so don't worry about that. But you should know, if you see Ig something, it means antibody, right? So that's important. So D, that's the fourth part, are of course the cells. And I showed you this slide before, and I said we come back to these lymphocytes. So lymph, right? It's found in the lymphatic system, and these lymphocytes are part of the adaptive immune system. So those were the cells, I think I told you, know these ones. And so now we're going to talk about lymphocytes. So there's some pictures of these lymphocytes. You can see it's got a really large nucleus. I don't really know why that is. It probably has something to do with antibody production, but that's what these lymphocytes do. They produce antibodies, and lots of them. So there's actually two types of lymphocytes. There's the B cells and the T cells, or the B lymphocytes and the T lymphocytes. And they have a special type of antibody that's bound to their membrane. And they might secrete antibodies, at least the B cells do anyway. So you can see why they're called B and T, right? The B cells mature in the bone marrow, and the T cells mature in the thymus, so B and T. I'll talk about what mature means in a couple of minutes. But like I said, these are antibody-producing cells, and of course that means these cells produce something that's going to bind the pathogens. And they're also involved in the pathogen killing as well, but they're going to do it in different ways. And for some reason, they're also matured in different parts of the body. So let's take a look at these B cells and these T cells. So first thing to know is about this maturation thing. This actually has a technical term in the scientific community called clonal deletion. So clone, like I said, are copies. And so these cells, they grow and they copy each other, and then they kind of differentiate. And these different cells will make different antibodies. And it's actually a very random type of process. And so you got to imagine, if you're making random antibodies, some of those antibodies may actually bind human cells, and this would be terrible. You do not want our immune system to attack our body. So they need to be, the ones that are going to be autoimmune, or they're going to react to the human body, need to be destroyed. And so this is called maturation. And they do this in different places, the thymus or the bone marrow. So like I said, you kind of have these juvenile cells. They start growing and they start recognizing things, and there's a mechanism in the bone marrow and a mechanism in the thymus to delete or to eliminate the ones that would react with self. Of course, this doesn't always happen, and there are autoimmune diseases, but for the most part, this is working really well because, like I said, it would be catastrophic if we didn't have this mechanism for clonal deletion. Another term is something called clonal selection, and this is what's going to happen during our immune response. And this is where one that you're going to have, like I said, a whole bunch of random T and B cells, and sometimes there'll be one that'll recognize pathogen. And that's the one that's going to get selected for. That's the one that's going to get enhanced and going to form both an immune response and an immune memory. So we'll talk about how that works in a few minutes here. And this usually happens in the lymphatic system, so lymph nodes, which is why when you have that infection, the cells are multiplying and there's a lot of activity going on. So there are lots of types of lymphocytes, particularly with the T cells. It gets really, really complex. And I'm not going to expect you to know about all of these different types of lymphocytes for this class. We're mostly going to make the distinction between the B cells and the T cells. But it's worth it to know that there's a whole bunch of different types of T cells. You can see they have different roles. We have, I mentioned the natural killers. These are the ones that don't need help from other parts of the immune system. They can just naturally kill things. Some are, not natural, like the cytotoxic T cells you can see there. It will kill cells with certain antigens, but it needs a little help. It needs to be activated by other cells, and so these other cells you can see are called helper T cells. So helper T cells are kind of like office managers, where they tell everybody what to do and give messages and emails and faxes and those kind of things. The messages are called cytokines. We'll talk about those shortly. And there's a whole bunch of regulatory ones. And there's other types of T cells. Like I said, there's a big long list. But we want to mostly in this class make the distinction between the T cells and the B cells in terms of what they're doing. But do know there are many types. So the last thing are these chemical signals and mediators. And again, this is something that includes cytokines. And these are chemicals that will activate or tune down the immune system. Sometimes, we don't want the immune system going crazy, right? Sometimes we have to actually tune it down and turn it off, because otherwise we're going to cause damage to our own body. There's a whole bunch of different types of these. You may have heard of some of these names in your travels. Interleukins, some of them have fancy names. TNF alpha, which I can't even remember what that one stands for anymore. Or IL-2, IL-4, and they all have different roles in the immune system. Some of them are used as therapeutic agents. When people have different types of cancers, they may go on interleukin treatment, which is designed to try to ramp up the immune system to try to take care of the cancer, right? So I'm not going to ask you anything about cytokines or interleukins or these messengers on the exam. It just Again, it's just an extra layer of complexity that we don't need and we can't really cover in one course. I will say this, though, that this is something that's been in the news a little bit due to COVID-19. They talk a little bit about people and these cytokine storms. So I think I'd mentioned it before, is that with COVID-19, people who have severe illness, usually in the first week, they're getting respiratory symptoms from the virus multiplying, and by the second week they're starting to get other symptoms, and some people are getting fluid in their lungs and things like that, and this is from the immune system kind of overreacting, and this is called a cytokine storm, and in some cases it can be quite fatal. So what is going on here? You've got these chemical messengers, and they're activating the immune system, and that causes further activation, which causes further activation, and so on, the point where it's bad for the body. We don't really fully understand why this happens in some people and not in other people. And this was the case with back with the H1N1 flu in 2009. There was a case, it happened like about a week before the vaccine came out. And it was in Ontario, and there was a 13-year-old boy. And of course, the media talked about how he was quite active and a hockey player, and he got the flu and dropped dead 24 hours later. And this is what they think happened. He had a healthy immune response, a little bit too healthy. And one analogy of a cytokine storm is that it's kind of like a runaway freight train, right? The immune system is just going and it just can't stop. So like I said, this is something we don't fully understand why it happens in some people and not others, and can be quite dangerous. But not going to ask you about it on the exam. Okay, so let's talk about the humoral response. I do have a video that I want to play for you. Let me just see how we are. Okay, we're doing great for time. But you can see we're going to talk about two immune responses that are part of the adaptive immune response. First one is the humoral response. So humoral doesn't necessarily mean funny. Humor kind of a couple hundred years ago medically meant blood, really, quite honestly. Right? People talked about the humors and the different types of fluids in the body. And so this means a response in the blood, and really it's going to involve B cells mostly. And the B cells secrete antigens, and the antigens are going to B cells secrete antibodies, and the antibodies are going to bind antigens and do their thing. The other is called the cell-mediated response. So that means we're not doing things in the blood necessarily. We're targeting bigger, usually larger objects that can't be destroyed by phagocytosis. And the T cells themselves don't secrete the antibodies. They just have the antibodies on the surface. And so they act in a little bit of a different way. So what I want to do is play this video for you. It's only a few minutes long, but it's actually a pretty good video showing the humoral immune response. So what I need to do now is just change and share on YouTube and share my sound, and I'll play that for you, and then we'll talk about it. So here we go. Hopefully you can see that. I'm just gonna make it full screen and press play.
In 1960, Sir Frank Macfarlane Burnet was awarded the Nobel Prize for his contribution to the field of immunology. Burnet's clonal selection theory revolutionized our understanding of the immune system. To this day, Burnet's research remains the foundation of the way we think about disease and how the body responds to it. Clonal selection is the central mechanism by which your body fights infection. The following visualization presents clonal selection during an infection by strep throat bacteria. This patient has a strep bacterial infection causing a red, sore, pussy throat. The tiny round strep bacteria thrive on throat tissue. The bacteria are coated with hairs called M protein. M protein is what your body recognizes to be foreign. In response, your body will launch an immune attack against it. Your lymphatic network is an important part of the immune system, detecting any infections that arise in your body. The M protein hairs drain into the lymphatic system around this patient's neck. The lymph node is crammed full of B cells. B cells are guardians of your immune system, scanning the body for foreign material. Arriving at the lymph node are M protein hairs from the bacteria. Your body contains billions of B cells. Each B cell is unique and is sensitive to only one kind of foreign infection. By chance, one B cell is sensitive to the M protein hairs from the bacteria. This is the molecular surface of that B cell, magnified 10 million times. Its surface is covered with receptors that recognize M protein. The receptors grab onto the M protein hairs. This triggers an activation signal to travel into the cell. The activated B cell migrates and begins to replicate. In a few days, thousands of copies of the selected cell are made. This is the central idea in Burnet's clonal selection theory, where one cell is selected out of billions of B cells and identical clones have been created from it. Therefore, the cell is clonally selected. This creates an army of B cells that mass produce M protein antibodies and release them into the surrounding fluid. The antibodies circulate in the blood and travel into tissues. They bind to the M protein hairs on the strep bacteria. This flags them for destruction by the immune system. Other immune cells ingest the antibody-covered bacteria. They destroy the bacteria and eliminate the infection.
Okay, so pretty cool video. I thought that guy, cool accent, of course. He does a great job of explaining what's going on, and you can see these principles we've talked about, right? So antibodies being produced and targeting something, and the clonal selection, and all those kind of things going on there. So it's not the only way that the humoral response works. Like I said, the immune system is quite complex. There are other ways that it may be activated. We mentioned those dendritic cells last day as a mechanism for recruiting the adaptive response. So sometimes they are part of the adaptive response as well. But you can see a good example there with the strep throat. So this is kind of my little flow chart, and like I said, just keep in mind this is a very simplistic version of the events. But a couple of things to point out, right? So the first thing is that there's an antigen, and like I said, there's different ways that it might get, the immune system may get alerted to it, but a common way is those dendritic cells. And I mentioned before that they're a little bit like watchdogs, and they grab onto things and hold on to them. And that's a chance for the immune system to kind of come along and say, is this self or is this something that's foreign? And so you can see that dendritic cells, I think I mentioned before, are sometimes called antigen presenting cells, and that they will do that. So the antigen comes along, and somehow it's going to get recognized by the immune system. And of course, this is going to happen by a B cell. And like I said, you know, there's going to be tons of different B cells, and there's a little bit of chance involved, but one of them recognizes it, and that's going to lead to its activation. So when it's activated, two things happen. One thing happens, it clones itself, and the clones do two things. The clones become active, and they're going to secrete antibodies, which is going to lead to the destruction of the pathogen. And some of those clones form memory cells. So memory cells are long-lived cells that are going to be sitting around and waiting for another exposure someday in the future. So we'll talk about that memory and immunity in a little bit here. So what happens when antibodies are secreted? The antibodies do their job, which is all of these things that we talked about. So opsonization, cellular toxicity, agglutination, neutralization. And all of those things ultimately lead to either phagocytosis, or they lead to cytotoxic killing, right? So they're destroying the pathogen. So that's good, that's what we want. So what about the cell-mediated response? I do have a video there, and I'm not going to show you this video. It's a very good video. It's just a little long, it's 7 minutes. And it's a little bit complex in that it's going to talk about a lot of things that aren't as important to go into detail in this course. Like, it talks a lot about the helper T cells, and it talks a lot about some of the cytokines and things like that. But if you have a little bit of time, it's worth checking out, and I'll see what I can do. I think I have an actual download of that, and I can probably post it to the Facebook group. And you can check it out. It's very good, and, but like I said, it's just a little bit long, and I do want to talk about other things today. So the one thing, like I said, about the cell-mediated response is that the targets are usually different, and they're usually bigger. So big things, obviously, you know, we're talking about things like worms. Of course, every time I touch the screen, it wants to pop away from me. There we go. So large parasites, right? So worms, obvious, you know, but there's lots of protists that are just too big to be consumed by phagocytosis. Infected cells, right? So of course, viruses will get inside our cells, and many of our own cells are too large to be destroyed by phagocytosis. But now that's an infected cell, and that's a liability. So the immune system wants to get rid of it. So a lot of responses against viruses actually do use the cell-mediated response, and in some ways, it's a more powerful response than the humoral response. Last thing is also abnormal cells. So by that, we mean cancers. And yeah, we are making cancer cells our body on a daily basis. And the good news is that we are eliminating them. Eventually, though, something might get through, which is why as you get older, you're more likely to get cancer, unfortunately. But yeah, our immune system does a great job clearing these things. All of these things are usually destroyed, like I said, not by phagocytosis, but by cytotoxic killing of some sort. And so it's a matter of recognizing them and killing them by toxins. So here's kind of the typical response. Like I said, we're talking about larger things. So we've got some sort of invader or cancerous cell or virally infected cell, and it gets recognized by a T cell. So these T cells are floating all around the body, and some of them, you know, they might recognize that antigen as something that's not normal, not human, not self. So the T cell gets activated. Like I said, with T cells, it's a little more complex. There's these helper cells and these regulatory cells that are kind of involved in the whole process. The helper cells, by the way, are what are typically infected by HIV, which can totally cripple the immune response, so they're very, very important. And then the T cell clones itself, so kind of like the B cells, but it doesn't secrete antibodies. It becomes cytotoxic, which means it will kill things by that kiss of death mechanism, so injecting toxins. And then a bunch of them will become memory cells as well, so long-lived cells that are there for a future immune response. So a little bit of a different system going on here, but they do complement each other, right? So smaller particles like viruses can get killed by the humoral response. The virus gets in a cell, and then that infected cell can get killed by the cell-mediated response. So there's a lot of complementing of these systems going on. So there's kind of a little diagram comparing the two. You can see humoral immunity. We're looking at usually smaller extracellular microbes, and the response is releasing antibodies. There's my little note there, and of course this has a lot to do with B cells. And the cellular immunity is larger things. So we're talking about large pathogens such as worms, virally infected cells, and cancer infected cells. And of course, this is carried out by T cells, which they become cytotoxic and kill the thing directly. So this is kind of just a little bit of a summary showing, you know, that there is a bit of interaction between these things. You know, they all have antigens. Sometimes they'll involve macrophages or dendritic cells. And helper T cells do activate B cells as well, by the way. Like I said, there's a lot of crosstalk between these parts of the immune system. And I'll show you another diagram here. This is from the textbook, just showing you that, you know, there's a lot of communication going on, a lot of complex interactions that make the immune response very effective, which is good. If you think about the immune response, you want it to be active when needed and not active when it's not needed. And you don't want to be too active, but you want to be active enough. And so the regulation is very, very complex. So this is from, I think, the first edition of the textbook. I don't know why they did not include it in the second edition. I kind of like this chart because it's showing you that different parts of the immune system might get activated to different types of pathogens, right? So if you take a look, I'm looking in the middle, right in the middle we have extracellular bacteria, and we're mostly looking at antibodies. You can see IgM, IgG, IgA are all involved in attaching and binding and killing those bacteria. If you have intracellular bacteria, so they're infecting the cell inside, secreted antibodies aren't doing anything at all, which is why we need cellular mediated immunity of the T cells, right? Viruses, you got a little bit of both. Kind of depends on the virus. Some viruses don't activate the IgM. I don't know why that is. So you got all this complementation of the immune system, which is kind of cool. I'm not sure what the red and yellow are representing. I can't remember why they have those bars there. Okay, so I want to switch gears and talk about memory cells and eventually vaccination, which is obviously super important. Um, so memory cells, of course, are these long-lived cells, and I mentioned that we don't really know why some types of memory cells seem to live a long time and others a lot shorter. But they are a good thing. And these memory cells, you can see this note here, it says they respond immediately and they will clone themselves rapidly. So let's take a look at kind of two infections. A primary infection, so that's the first time you get exposed to a pathogen. You can see pathogen here. There's an exposure, and there's a little bit of a lag. It takes time for that randomness, that random B cell or that T cell to find the pathogen and react and clone itself and get activated. So anywhere from 3 to 5 days, and then we start producing antibodies. So IgM is produced first, and then IgG, and so usually within about 2 weeks, you've had a relatively strong immune response, and hopefully you have not died. And then the disease runs its course and hopefully goes away. So small amounts of antibodies are produced. The second time that you are exposed to something happens really quickly. These cells are primed and ready to go, and in many cases, you don't even know you were exposed. In some cases, you still get infected, but maybe you don't get sick. In some cases, you don't even get infected at all. So like I said, the measles, immunity to measles is so amazing, you don't even get infected. Other diseases, you might get infected and have a small infection that's detectable, but there's no disease. And like I said, this happens not days, but hours. These cells, they get activated, they clone themselves, they secrete their antibodies very, very quickly to the point where it's very measurable in the blood. So if you've been exposed to something, you can get something called immunity. And immunity means that, hopefully you're not going to get sick in the future. And so I want to talk a little bit about immunity, and there are different types of immunity that we can get. And, like I said, this is going to lead to us talking a little bit about vaccination here. So there's types of acquired immunity. So you can see I have a list here. Natural immunity is the one that probably makes sense, right? You get the infection. So you can see this child here has the chickenpox, and the disease runs its course. The immune system makes antibodies, clears up the virus, and then the memory B cells are there, and the child probably will not get chickenpox again. Maybe by the time he's 60, he might get shingles. And that's when those memory cells are starting to decline and the immune system is not as strong and vigorous. But we're looking at, not quite a lifelong immunity, but pretty long for chickenpox, which is incredible for most people anyway. So this is kind of, there's lots of people talking about this for COVID-19, and I'll kind of address that in a minute or two. The question is, for COVID-19, we don't know how long natural immunity is. Some people think it could be 6 months. So what does that mean? You're getting an infection every 6 months? It could be years. We don't really know. We've only had this virus around for like, less than a year. So we just don't have the data yet on this kind of thing. So this used to be a strategy, by the way, way back before there was a chickenpox vaccine. Some people would have chickenpox parties to try to give their child immunity to it, because getting chickenpox as a young child is usually not so bad. Sometimes there are complications, so there were risks, but kind of the majority of cases, people get a few blisters and then it runs its course and that's it. Getting chickenpox as an adult can be a little bit more severe, so it was always a good thing to get it as a child in some ways, and then you'd have your immunity. And so this was kind of a thing. And somebody's making a comment about people still doing this. So that's, I'm showing a lollipop there because there was somebody a few years ago trying to sell chickenpox laced lollipops on Facebook. And I think they got arrested for it, which is good. I would not recommend this necessarily. Like I said, the risk is higher. You know, there are some serious complications, right? And you know, I mean, it is a strategy, but I think the risk is higher than getting the risk for the vaccine. I know when I was a student, there was a measles case in my child's school, and people were talking about having measles party, and it was just like my mind was blown. Measles, like chickenpox, is one thing. Measles is a very serious disease that can decimate your entire immune system, and it was like, holy cow! Like, what are you talking about? Are people, you know, they don't know what they're doing. It's unfortunate. And there's been this kind of thing in the news about COVID-19. You know, in the States, I can't remember where it was. I think it was Georgia. There were a bunch of students that were having COVID-19 parties, and some of them got seriously ill from it, and some of them mildly ill. So like I said, there's a lot of risk in this kind of thing. But, you know, it has been historically a strategy, and for some reason people think it's a good idea now. I don't think it's a good idea. A couple people have some comments about, yeah, about somebody's mom getting PJs from friends whose sons had it, kind of thing. Yeah, and like I said, this used to be a strategy, and before the vaccine, and you know, it was a lot better getting it when you're like 5 years old rather than getting it when you're 25. So, you know, anyway, I don't recommend it. And I hope that other people do not as well. Trying to combat misinformation here. So if you could do that as nursing students, that would be wonderful. So there's also natural passive immunity. I mentioned this already. The mother can give antibodies to the child through the placenta and also through the breast milk. This doesn't work for every infection, but a number of them. And you know, this is something that these antibodies are in the blood and will live in the child for some of them up to a year. So this is great. It means that a child can have some immunity to certain infections for about a year after birth and pass over from the mother, which gives lots of time for the baby to start developing its own immune system, which is great. So artificial immunity, that's vaccination. So the whole idea here is we're exposing people to something that's either similar or similar enough to the real thing to get a good immune response, so that when we are exposed to the real thing, we're not going to get sick. There's a picture there of a little boy getting the oral polio vaccine, and so we'll come back to vaccination in a minute or two. So there's one other type of sort of passive artificial immunity, and it's been in the news again lately, and this is where we're using some sort of harvested antibodies for therapy. So a classic case is something called antivenin or antitoxin. So if you live in like Medicine Hat area in Alberta, you may know that there are rattlesnakes down there, and rattlesnakes are poisonous. And so the emergency ward there would have some antivenin. And how is this done? So basically, it's done often in horses, by horses, because they're large mammals, and that means you can take a liter of blood and the horse isn't going to miss it. And so they inject, they started with very, very small quantities of the toxin and inject a little bit in the horse, and then they build it up and they inject more and more until the horse produces a very strong immune response. They extract some blood, and from the blood they isolate the antibody, and they have it there sitting there, and if somebody does get bitten by the snake, they can stick it into their blood right away, and it's going to hopefully neutralize the toxin. And it can be very effective if it's given quickly enough. And so there's a whole bunch of these things, and depending on where you are in the world, your emergency room may have them or may not have them in the emergency room. Some of these things now, we're at a stage technological wise that some of these things we can actually make them artificially, and so I'm kind of wondering, you know, when we're going to start to see those used therapeutically. And there have been some that have been experimented with COVID-19. And so here's this is a different term. It's called convalescent serum. So that means the serum can come from somebody who was already sick, or it can be artificial antibodies. So the whole idea here, this has been done, by the way, with some infections like Ebola successfully. Problem is usually you need a patient with antibodies. So you can see here's this red guy, somebody who got sick. Let's say it's Ebola, and they recover. And of course, shortly after they've recovered, particularly within that 2-week period, they're going to have a lot of antibodies in their blood. So you say, hey, can I have some of that blood? And you grab some blood, and then you give it to somebody who's sick, and it might give them protection. So this was done successfully in a couple of patients with Ebola. Like I said, the unfortunate thing is usually you need somebody who's been sick, and you need a lot of blood, but it's kind of an emergency kind of medicine. So there's been some tests now with COVID-19 where there's been a company or two that have been making artificial antibodies and wondering whether this will be a good type of emergency type of medicine. And apparently, I think this was actually used on Donald Trump when he caught COVID-19 as one of the therapeutic techniques. So it's kind of an artificial way you're giving somebody antibodies made by something else, whether it's a horse or another human or done artificially. So let's talk about vaccination. I like our textbook for this course, but this is one area that it kind of fails, I feel. It does talk about vaccines. It's not an immune system part of the textbook. It's kind of another part of the textbook, and it's kind of too short for my liking because they're very, very important. So vaccination is sometimes called immunization. So the words are used kind of interchangeably. And like I said, the whole idea is you're trying to give someone immunity to a pathogen by giving them something that kind of looks like the pathogen. And there's different strategies to do that and hopefully form memory cells. So I think I showed you this slide way back in the first lecture. This is where the word vaccination actually comes from. So this is Edward Jenner, and he was, I think he was a surgeon, and he was, working in the countryside, and he had heard about these milkmaids. So milkmaid is a woman who milks a cow, getting cowpox, and that they never seemed to get smallpox. And he kept hearing about this, so what do you do when you're a doctor or a scientist back then? You test your hypothesis on a small boy. Yeah, I mean, maybe the ethics weren't there, but basically what he did is he exposed this boy to cowpox, and a couple of weeks later exposed him to smallpox, and the boy was immune. And so he published about this, and this practice became widely spread and eventually led to the eradication of smallpox. So the word vaccination, vacca, is actually Latin for cow, and so that's where the word vaccination comes from. So this is a very fascinating story all about Edward Jenner. Like, he didn't just do this immediately. He actually researched this for like 15 or 20 years before kind of doing this experiment. So he was pretty sure it was going to work, even if the ethics weren't entirely there. So what is a vaccine? Like I said, it's a preparation that is going to hopefully give the person a strong immune response and hopefully good memory cells, right? Which are going to give immunity against that disease. There are different types of vaccines. There's a whole bunch of different types. I'm going to talk about actually 6 types. There's 4 types I'm going to talk about right now that are currently used in human vaccines. And you're probably familiar with some of them. And they mean different things. So let's kind of discuss these. First one is attenuated vaccines. And so this is kind of, probably I would say the oldest technique for making vaccines. It was done by Louis Pasteur, and for making rabies and cholera vaccines. And the whole idea here is you use an altered pathogen, right? So the pathogen can be genetically altered. Nowadays, by modern techniques, it's pretty easy to do. Classically, what they would do is they would grow and regrow and regrow and regrow a pathogen in the lab, sometimes exposing it to radiation, hopefully weaken it. So the pathogen can infect a human, but hopefully only grow really slowly and not make the person sick. And these will actually give you a very, very strong immune response, because the pathogen can sit there in the body, sometimes for years, and sort of, priming that immune response. And so a lot of these actually are very, very good vaccines. And you can see some examples there. We have the oral polio vaccine, OPV. We're going to talk about that polio vaccines in a few minutes. The MMRV, so the mumps, measles, rubella, varicella vaccine is actually 4 attenuated viruses. And there's also the flu mist, the nasal flu shot. So the one that's not a shot, I guess, the one that's getting sprayed up the nose is also a attenuated vaccine. So sometimes we use killed vaccines, so we're killing the organism or deactivating it somehow. There's different ways to do that. You can use radiation, or you can use chemicals. Or you can boil it. There's a whole bunch of techniques. Usually they're using chemicals nowadays to kill the organism. And so there's another polio vaccine. The one that probably most of you got is the injectable polio vaccine. That is a killed virus. Rabies vaccine, the usual flu shot, is also a killed or inactivated virus as well. So sometimes we don't need to use the whole organism. So this is kind of the third category where, it's called, often they're called conjugate vaccines or subunit vaccines, where we're just like, why inject the whole organism? So you can inject a protein or a carbohydrate or something like that. So you can see my example there, the pneumococcal vaccine. So there's one called Pneumovax 19. I think the newer version is called Pneumovax 23. And it has 23 carbohydrates. And those carbohydrates are the exact same as the carbohydrates that are found on the streptococcal organism that causes pneumonia. So I think they're not even made, I think they're made artificially too by chemists. So those carbohydrates will give the person an immune response. You don't need to inject the entire organism. HPV vaccine, hepatitis B vaccine, conjugate just means it's usually bound to something else to help boost the immune response. And you can look that up if you're interested in more details on that. So the last category is kind of similar. These toxoid vaccines, this is some sort of inactivated toxin. So I mentioned when we were talking about toxins, exotoxins, some of those exotoxins are AB toxins, right? So the A part is the thing that does the damage, and the B part is the thing that allows it into the cell. So the diphtheria toxin is exactly that. It's just half the toxin. So you actually have an immune response to the one half of the toxin, and it's good enough to neutralize that toxin so that it can't do any damage. The tetanus toxin vaccine, I'm not as familiar with how it's formulated, but it's some sort of inactivated version of the toxin, something that can give a strong immune response, but and prevent disease, but not causing any damage to the human cells. So here's kind of a list of a bunch of different vaccine types that are used in Alberta. And some of these are not routine vaccines, like hepatitis A. You're probably not going to get a hepatitis A vaccine unless you're traveling to a place where you might encounter contaminated water. But yeah, most of these, I think I've got a pretty complete list of the routine vaccines here. If you're interested in what's going on, you can see that they're using all sorts of different strategies. Nowadays, often you can see, like with the MMR vaccine or the DTaP vaccine, we're actually including multiple antigens in one shot, which makes it a lot simpler. So we're not going in, bringing our babies and our children in, every week for the rest of their life. Make things a little more simple. One thing I was going to mention is the inactivated vaccines and the toxoid and subunit vaccines. They are more likely to need a booster shot than the attenuated. And that's because the attenuated, like I said, it's usually sitting around for a while, replicating. Some of them do require booster shots, but not as common as the other types. So I wanted to show you some data here. I found this this article. It was published a few years ago, I think in 2006 or 7, but just showing the effectiveness of vaccination, right? Like if you take a look at, they had it categories into 2 parts, right? Vaccines before 1980 and after, right? But, I think this is a very fascinating list, right? I'm looking at this list at the top here, and, I'm looking at this list here, And I have never in my lifetime seen any of those diseases, which is just mind blowing. Now, a little bit before me, I think one of my sisters had measles. So I don't know when the measles vaccine came out, and my sister-in-law had the mumps. And I have a good friend who had pertussis, but I have never encountered those diseases. So they're practically wiped out or eradicated, although some of them are making comebacks due to people not getting their vaccines. Measles was eradicated in Canada in the year 2000. Is that hard to believe? That's amazing. Measles was eradicated in North America. And now it's now we're looking at 200 to 300 cases a year in Canada. It just breaks your heart. Some newer vaccines you can see down here since 1980, right? So I think that what it's showing here on this list is that the ones that were introduced a few decades ago, we're looking at a very high percentage of elimination. The other ones, we still have some headway, right? These ones below, I'm looking at them, and I've only had 2 out of those 5 vaccines myself, because probably they weren't available when I was a child. So let's talk a little bit about herd immunity. This is something that's popping up a lot lately with COVID-19. And people want to, some people think, why do we need a vaccine? Why don't we just wait till we get herd immunity? So let's talk about what that is and why that might be a problem. So if you take a look at this, you've got a naive population, so a non-infected population, right? Very few people are immune. Somebody gets sick. All those people that are gray are likely to get sick because they're in contact with that person. The whole idea is more and more people get sick, more and more people will in theory get immune, and then it's less likely that a sick person will be in contact with someone who's susceptible to the disease, right? So you can see in this last chart here, we've got a very high percentage of the population immune, so the illness can get basically eliminated. So why is this a problem? Because generally, we don't see this kind of thing happen as an illness runs its natural course, right? This kind of thing, diseases just keep propagating and propagating. We keep having babies, we keep encountering new people. The only way that we know to get good herd immunity is to actually have a very high vaccination rate. It depends on the disease. For measles, measles is very, very, very infectious. So we need about 95% of the population to be vaccinated to prevent measles. Other diseases like influenza, it's only about 50%. If half of Canadians got their flu shots, we would probably not have a flu season. So it kind of just depends on the illness. Most estimates are with various infections are about 70-75%. But it doesn't happen normally naturally. The one exception is really, really dangerous diseases like smallpox. And we see that historically where smallpox would get to a city, wipe out a whole bunch of people, everyone who was left was immune, but it would come back a few years later once people started having more babies or new immigrants and whatnot had come in, right? Because it just sort of would ebb and flow. But in that case, we're looking at a very highly virulent disease that spreads very quickly, and we're not looking at the case with most diseases. So I see I'm running out of time. I talk about a few more things. I was hoping to finish this today and talk about COVID-19 as well a little bit, but that's fine. We'll spill this over to Tuesday or Thursday. Thursday's lecture is a little shorter, so we should have time to finish this off. But I want to talk about a couple of new types of vaccines that are making the headlines because they're in the news dealing with COVID-19. So this is something that has been researched for a few years now in animals, and these things called nucleic acid vaccines. So the whole idea here is rather than inject, let's say, a viral spike protein, let's just inject RNA or DNA that codes for that protein. So the whole idea is you inject the RNA, let's say, and the RNA gets into your tissues, and your tissues read the RNA by the ribosomes, and they make a viral protein, and then your immune system can have a response. So it's kind of a cool idea in some ways, and it turns out that it's super easy to manufacture. The distribution may be a little bit harder, but the manufacturing, it turns out, is super easy. So there were at least a few companies researching this right before the pandemic, and they literally finished all their animal studies and were ready to go into humans. And so due to the ease of manufacturing, these companies are kind of ahead in the whole vaccine race for a COVID-19 vaccine. So we'll talk about that probably, I'm guessing we'll probably get there next day with the COVID-19 vaccine race. We'll see how much I cover here in the next 3 minutes. There's another type of vaccine that the same thing has been researched. And actually, there had been an approved Ebola vaccine using this technique in humans, but no other normal routine vaccine had used this. These are these viral vector vaccines. So the whole idea here is that you get another virus, so maybe something that can't replicate in humans. So maybe you get one from a different animal, which is done. These adenoviruses, you can get them from all sorts of animals, or a weakened virus. So you can see the example there is a weakened measles virus. And what you do is you genetically engineer this so that this virus-like thing expresses the new virus's proteins on its surface. So why would you do this? Well, like I said before, the larger the object, the more likely the immune system has a chance to recognize it, right? And so if you have something that is already known to attract immune cells, that's going to be your delivery method. So it's just another delivery method to get the antigen into the body and hopefully attract those immune cells to get an immune response. There's a vaccine being developed right now in the UK against SARS-CoV-2, and they're using a chimpanzee adenovirus. And I think it was initially called ChimpVax, and then they realized they didn't want to necessarily advertise they were using, chimpanzee and chimpanzee jokes and all those kind of things. But it was a virus that didn't grow in humans, so it was actually a great vector. And something that was known to give a good immune response. So maybe I will address this question, and then next day we'll talk about COVID-19. It says here, why is there not a vaccine against cold viruses? Well, it turns out that the common cold is caused by at least 200 viruses. There's, I think, I'm trying to remember what the number is, 130 maybe different rhinoviruses that cause the cold. They mutate from season to season. Never mind those other types of viruses such as adenoviruses, coronaviruses, parainfluenza. These can all cause cold-like symptoms. So it's quite complicated, right? And the other reason is, quite honestly, people are not usually dying of the cold. It's an inconvenience, and they're super annoying, but the challenges are huge. So that's why there was not a ton of coronavirus research up until recently, because of course most of them were mostly annoying and not life-threatening. So it's one of those things that we kind of in retrospect wish we had done a little bit more about coronaviruses now that we're in the middle of a pandemic. So I will just finish off on this slide, and we'll come back to this next day and just say that for COVID-19, we're trying all the strategies. There are at least 200 candidate vaccines out there. So we're trying these nucleic acids, these viral vectors, we're trying attenuated viruses, inactivated viruses, protein subunits, and there's probably a few more that aren't on this nice diagram that somebody in Nature made. So we'll come back to this next day on Thursday, and we'll talk a little bit more about COVID-19 vaccine, and I also want to talk about the polio vaccines, and then we'll do topic 13.3 as well, which is immune disorders. So see you next day. Hopefully you enjoyed this lecture. I think this stuff is very, very fascinating, and please let me know if you have any questions. Have a wonderful Tuesday.